that. Thank you, Amit.
Morning, and thanks again to the Department of Medicine, Dr. Tambu, for the kind invitation. So I'll be speaking on gut microbiota. Now, gut microbiota actually is a very extensive topic. You know, it obviously is difficult to cover all of it in an hour, but I'll just give you an overview of the structure, function, and what are the clinical implications of gut microbiota. Next slide. So essentially, I mean, in the past, the study of microbiota was dependent on culture actually, and most of the organisms in GI tract, anaerobes, were very difficult to culture. So we were not able to appreciate what is the true diversity, what is the true abundance of organism in the past actually. But now with the availability of molecular tools, we are now realizing, how many different types of species and organisms are there, what is their diversity, what is their abundance actually. So this whole landscape of microbiome has changed after the availability of these molecular tools. And actually there is dedicated consortium, like you have for the genome, you have the Human Microbiome Project from US, you have the MetaHit, which is from the EU, the Canadian, Australian, all different groups have come and formed consortium. And as a result of this group activity, we know much more about the microbiota than we knew about 20 years ago. There's also one Indian microbiome project which is right now underway, led by somebody from Pune, and few people are involved in that. So we'll also know in some time about what is the microbiome thing and these projects look at all parts, not only GI tract, the skin, the respiratory, all tract they'll be looking at actually. So as a result of all this, you could see that the number of publications in the microbiome has really shot up, because now the microbiota, the dysbiosis has been implicated in all conditions, not only GI. Almost every system you think of, the disorders have been implicated as a microbiome as a cause of condition. So therefore, there's tremendous interest in microbiome, and as you can see, the number of publications have really shot up dramatically in the last 10 years about the microbiome. So before we go into my talk, just 2-3 terms which are used very frequently when you read a literature on microbiome. So what is a microbiota? So microbiota is essentially all the microorganisms which is present in a defined environment. So if it is a GI tract, it's called gut microbiota. If it's respiratory, it's a airway microbiota. Whereas microbiome means actually genetic material which is present. But often in general thing, these two terms are used interchangeably. So when you say microbiota, microbiome, people generally mean the same thing. But technically, this is what these two things mean. And dysbiosis, which is actually the key word we use when you talk about disease process. Dysbiosis means that the microbiota in your particular area, but I need to see it also. So in that particular area actually, there is a change in the structure and also a change in the function of organism actually, okay? So that's what dysbiosis means. So this is how I have divided my talk today. We'll first talk about the structure and the function of microbiota, how it develops, then function, then what are the tools right now available for us to study the microbiota, implications in disease, and finally end with some clinical applications. So when we say microbiome, normally I think most often we mean bacteria, but remember there is a whole host of other organisms as well which we have in our body, which includes your fungi, parasites, viruses. But somehow most of the research focus has been on bacteria, but remember we have a lot of other organisms, and I'm sure virus will rank second after bacteria in terms of the thing. They may be even more, because bacteria have this bacteriophages as well. So we really don't know, but viruses may be actually quite a dominant component. So microbiome includes all these organisms. And also any part of the GI tract, any part of our body which is exposed to environment will have microbiome. So whether it's your genitourinary tract, skin, eye, oral cavity, airway, GI tract, microbes are present in all these areas which is exposed to external environment. But among them, the bulk of them is present in the GI tract. So more than 60% of our microbes in our body are actually in the GI tract, and that's because GI tract has a large surface area, almost close to 200 meters square. So plenty of space for them to colonize and live in the GI tract actually. Now with so many different types of organisms, it's difficult to know the names of all of them actually, and we know that bacteria, we start from phylum, class, order, family, like that we come down. And among the phylum, but luckily among the phylum, the 3 or 4 phyla make up more than 90% of the organisms we have in our body. The most common two are the firmicutes and bacteroids. So these two make up the bulk of the bacterial phyla in our GI tract. Then you have proteobacteria, fusobacteria. Now these terms you'll keep hearing or keep reading again whenever you read a paper on microbiome. Okay. So these are the major phyla present in our gut. Coming lower down up to the level of species, at the level of species, it's now predicted that there are about 1,000 species in each individual, and majority of them are anaerobes. And that's the reason why these are not very easy to culture, and that's why culture was not able to appreciate the true diversity and nature of the organisms. But out of these 1,000, about 40 of them make up more than 99%. So about 2 or 3 phyla make up most of the bacteria, and about 40 species make up most of the, at the species level, most of the bacteria. And few examples of the common species I have cited: the bacteroids, Clostridium, Ruminococcus, Faecalibacterium, etc. Now, so if you look at the amount of genetic material which this bacteria bring to our body, they are significantly higher compared to what our own number of genes are actually. And so it's almost like an organ system. You have a structure, you have a gene pool, they have their own set of functions actually. And now the concept is emerging that even though the actual bacteria may differ from individuals, but the functional pool of genes which they have may be actually similar. And obviously all these figures may vary. So new publications keep coming, they'll dispute the previous figures. So I'm not really trying to give you an accurate idea about what's happening, but just to give you an idea about the magnitude of this, of the gut microbiome, the genetic pool we have in our body of this organism. Next one. In terms of composition, again we won't go into details, but just to tell you that they are present in all part of GI tract, starting from stomach till the colon. But in stomach, because of the acidic environment, the organisms are lower in density, but as you go down in the small bowel, they increase. And colon has the highest concentration of organisms, actually, of the microbes, the bacteria. The species also, the relative composition varies as you go from top to the bottom. And there is difference not only from top to bottom, but even if you look at the cross section of a gut. And if you look at cross section of, say, a small bowel, the bacteria which is close to the epithelial surface is somewhat different from what it is in the lumen actually. So there is a cross-sectional difference as well. And this is important because when we study a fecal microbiome, the fecal microbiome looks at actually the luminal bacteria. It doesn't tell you much about what is on the epithelial surface. So for example, for our project, we are looking at both mucosa as well as stool in the same patient, because the organism, the relative proportion is different, and what people feel is that what is close to your surface may be having a more direct impact than what is away from the surface actually. Okay, next one. So that was a little bit about the structure of the how much organisms and what the phyla and the species we have. Then about the how does the microbial population develop in the human beings. So fetus gut is sterile and the colonization begins soon at birth actually. The initial bacteria are all derived from the maternal microbiome, the vaginal and the fecal microbiota. In the beginning, the bacterial diversity is low. The actinobacteria, proteobacteria probably are more important. But as time goes by about 2 to 3 years, the bacteroids and firmicutes, which are the dominant things in adult, they start sort of assuming dominance. By about 5 to 6 years, we almost have the microbiome which is present in the adult. And that remains stable up to your old age. And then in the old age, again, there is a bit of a change in microbiome. So the development first about 2 to 3 years is critical, and up to about 5-6 years, there may be some changes, then sort of it stabilizes. But the type of microbiome you'll have, there are a lot of things which affect the composition of this microbiome, which includes your diet, the dietary pattern, dietary intake, your genetic sort of composition, age, and use of antibiotics, which part you are from, the mode of delivery, breastfeeding, formula feeding, smoking. So there are so many factors which actually affect your microbial composition, and therefore that's one of the challenges in microbial studies, because when you're doing a microbial study, you want to say a cause and effect between disease and particular organism. But with so many factors coming into play, it's actually difficult to pinpoint whether what is causing the true sort of a change in the microbial population. And also this highlights that things like mode of delivery, breastfeeding, etc. has an important effect. So therefore children, infants who sort of have a LSCS or a formula fed, their microbiome is going to be different from what it is for the other group actually. So that was briefly about the structure of the thing and how the microbiome develops in our body. Next we talk about the function. Next one. Now there are several, so as I said, the microbiome is like an organ system in our body, and so there are several important functions of gut microbiota. The important ones being it has a very important role in development of immune system, maintenance of immune function, a lot of metabolic functions. Also it has a role in development of the enteric nervous system as well as function of enteric and central nervous system. Then resistance of the gut from colonization by pathogens also is an important function. Next one. So this slide actually is it's an important slide just to give you a conceptual framework of how a gut microbiota being in the gut is able to influence so many organ systems close as well as far away from GI tract. So if you look at the gut microbiota, it is exposed to a lot of luminal contents, most important being diet, your bile acid, the medications we take. And these are acted upon by the bacteria by different processes. There's fermentation, hydrolysis, etc. and you have an end product. Then bacteria make their own products as well. Then you have the bacterial components, just like the cell wall components, and you have the entire bacteria. So the metabolic pool, the bacterial component, the entire bacteria, all these things together interact with the epithelial lining. They also have very important interactions with the immune system in the GI tract and the enteric nervous system. And some of the bacterial products will also leak through into the circulation, and they will affect organs away, like in nervous system, liver, muscle, heart, etc. And therefore the bacteria and their products have interactions with the epithelial lining, the immune system, nervous system, and that's how they can affect their function, and they can sometimes lead to disease process. And also the components can reach far away by circulation and can have effect in other distant organs and can lead to disease process actually. Okay, so this is right now the sort of overall concept of how a gut bacteria being in the gut is able to sort of cause disease at a far away site as well as in the GI tract. Next one. This again just gives you a diagrammatic representation that how the products of bacterial metabolism can affect organs which are away from the GI tract. Next one. So of which I told you one of the most important function is actually the immunological function. And most of the immune cells in our body are actually localized in the GI tract, which are constantly exposed to the microbiota. So therefore it's very important that we have this tolerance to this commensal microbiota. At the same time, we mount an immune response to the pathogens actually. Now, microbiota has been shown to have a very important role in the development of this lymphoid tissue. So this has been seen from the experiments on the germ-free mice. The germ-free mice, if you see there, the lymphoid system won't develop very well. Their immune system won't develop very well. So therefore, it's known that the microbe is very important for a development of a healthy immune system, to get immune tolerance to commensals, and also to have pathogen response to pathogens. And it also promotes effective healthy barrier function in the GI tract. And this is possible because of the crosstalk which happens between the microbiota and the immune system, that what you get tolerance to, what you react to. Next one. And again, more recently what has been shown is that to have a healthy immune system, the type of organisms you are exposed to in the initial few, say, months of your life is actually critical. So for example, for humans, what they have shown that in the first 6 months of your life, the type of bacteria you are exposed to will determine your development of a healthy immune system actually. That's why again this formula feeding, cesarean section, use of antibiotic in children, all of them probably have implications in having allergy, asthma, rheumatological disorders later in life. In mice, this window is about 2 to 4 weeks. So this concept of this window of opportunity initially, and that is why the importance of having appropriate microbiome in the beginning is important actually. Next one. So just to sort of, obviously this is a large topic, but again some of the things which are present on bacteria which are recognized by body includes your lipopolysaccharide, flagellin, etc. So we have our pattern recognition receptors which recognize these components, which is a part of innate immune system. They stimulate immunoglobulin A production, and the healthy bacteria promotes anti-inflammatory cytokines and down-regulates the pro-inflammatory cytokine. Next one. So that was about the immune system, which is a very important role of the gut microbiota. Then also they synthesize a lot of stuff, of which the most important is the short chain fatty acid. All of us know that short chain fatty acid has a important anti-inflammatory role, anti-carcinogenic role. So these are made from the carbohydrates which are not digested in the GI tract. The bacteria mostly in the colon actually act on them, and they produce this short chain fatty acid, which is acetic acid, propionic acid, and butyric acid. Apart from that, several vitamins like cobalamin, biotin, riboflavin, etc., vitamin K are all synthesized by the bacteria. The bile acids, which is in the GI tract, also the metabolites are formed. And drug metabolism, again very important point. So this may partly explain the inter-individual difference in response to drugs, and now it's being realized in the chemotherapy in a very big way, that the differences, especially with the immune checkpoint inhibitors between different patients, may depend on the type of microbiome you have actually. So again, all these things are a part of the bacterial function. Next one. And again, just to give an overview that how the bacterial metabolites, either an excess of abnormal one or a reduction of normal one, can lead to disease in different part of the system. So a lot of metabolites have been implicated in disease like you have the allergic disorders, cancers, gynecological disorders, cardiovascular. So all these metabolites have a role to play, have been proposed to have a role to play in different disorders. Next one. Next is our microbiome-gut-brain axis. All of us would have read about the gut-brain axis earlier, about the IBS and all that thing. But now is the concept of microbiome-gut-brain axis, because microbiome actually has a very close interaction with the enteric nervous system, and also the microbial products has an effect on the central nervous system as well. And for this reason, microbiome has been implicated in Parkinson's disease, autism, depression, apart from IBS actually. Okay, next one. Another important function is colonization resistance. So we have a healthy colony of bacteria in a GI tract, which our body is tolerant to, and it prevents the healthy group of bacteria we have, it prevents colonization by the pathogens. And there are several mechanisms. Production of products like bacteriocin, etc. There are inhibitory metabolites also, and as well as there is competition for nutrients. And this can sometimes go haywire, like what happens when somebody gets antibiotics, and then you get the C. diff infection. And C. diff infection is one example where the microbiome probably changes because of antibiotic, and then it gives a chance for the organism to grow, and that's why one of the therapy for C. diff is actually fecal microbiota transplantation actually. Okay, next one. And the other functions as well, again because of lack of time we won't go into details. Next one. So just to summarize in terms of function, I think the immune system development and function is very important. So is the function, so is the synthesis of so many of the metabolites, nervous system, okay, and also colonization resistance. So that was about the structure and the function, and now I'll go about how to study these organisms. So many organisms in our body, how do we go about studying this microbiome? Next one. So as I said in the beginning of my presentation, that the majority of organisms in our gut are actually anaerobes, and with the culture-based techniques, they will underestimate the bacteria because most of them are anaerobes, they're difficult to culture. So therefore, the culture-independent techniques are better. Now, one more important point to remember is not only the structure, but we should also know about the function. And as of now, the focus has been mostly on the structure, what bacteria is present, but actually what will finally make a difference is the function the bacteria is capable of in the body actually. So we have to think of assessing both structure as well as function, but function assessment is actually much more expensive. It's still a bit of, in early stages in many things, but it's something which is sort of coming up. Next one. So this just gives you an overview about the different techniques we have to study the microbiome. On the left hand is the culture-based technique which I have been talking about. The problem is only a small number of organisms, less than 30% can be cultured. It's a low throughput. So in one sort of thing, you can have one sample in your each culture media, and it takes days to weeks for the results to be available. So among the molecular, we have the PCR panel, we have the Amplicon sequencing, and we have the shotgun sequencing. The PCR panel, in that, this is a quicker test. But here the thing is, you need to decide priori in your mind what organisms I am looking for. And those will be picked up if they are present actually. So this is more helpful in a diagnostic setting. It is coming up for diarrhea evaluation in a big way in many centers. You have a PCR with all the things for Salmonella, Shigella, even virus in the same panel. You get an answer to it. But that is not very good for an exploratory research. You want to know what other organisms are present. So for those, you have the Amplicon sequencing or you have the whole genome sequencing. In the Amplicon sequencing, we are just sequencing a part of the genome, which is cheaper than the whole genome sequencing. So we want to sequence a part of genome which has enough information to tell you what organism that gene comes from. And that is called a marker gene. So we have a marker gene which is present so each organism may have its own type of a gene sequence actually. So this Amplicon sequencing looks at the marker gene in the organism. Now these are really high throughput. You can actually process up to about 300 samples in a single run, unlike in culture where you have a single sample in each media, or in the PCR where you can do 20 to 30 samples. It takes about 2 to 3 days you get your result, but obviously it is expensive. Now, the limitation with only a small part of gene sequencing is you don't get much idea about the function. If you want to know the function, you have to sequence a large part of gene or the whole gene actually. So this limitation is overcome by the shotgun or the whole genome. So where you sequence a large part of gene or the entire gene of the bacteria. So here it identifies the organism. It's also high throughput. You can do more than 300 samples in a single run. This also results are relatively quicker, but it's more expensive than your Amplicon sequencing. But the benefit is that you also get an idea about the functional potential, because all the genes you know, so you know which gene has what particular function. But because of the cost, it's limited. So for the Amplicon sequencing, also in the past it was very expensive. The cost has been slowly coming down. Currently you can get one sort of sample sequencing for about 6,000 to 8,000 rupees, which is still expensive. If you think of say 1 to 200 samples, it's about 12 to 15 lakhs. But it's much lower compared to what it was in the past actually. And that's one of the limitations still maybe in Indian setting is the cost involved in this sort of analysis actually. Next one. Last one, I think it's done in hematology and few departments actually. So the thing, most of the time, Tambu, it's actually a gene which if you want whole gene sequencing, it will be, I don't have much, but it will be definitely much more. It will be much more than this thing actually. Whole gene we have not gone. May not be that much actually. See, the thing is, it depends on what organism. So bacteria, DNA obviously is manageable compared to other organisms, but still it will be much more than what it is for this thing. So I think even see, most of the gene work is right now targeting at some disease-known genes or a 16S gene. The whole gene thing is mostly a research thing as of now. So that's why I think maybe people may not be doing it. But this can be done in the platform which we have in GI Science. We can do the whole gene sequencing, but somehow we have not done it. We're not required to do it for the project purpose actually. Okay, next one. So again, when you say microbiome, the most of the work has been focused on bacteria. So in bacteria, so what is the marker gene in bacteria? So this is a very important word to remember for the students when they write, say, question on this exam. The 16S rRNA gene. So this 16S rRNA gene actually is a marker gene which is used for studying bacterial community. And now what is special about this? So this is a 1500 base pair length gene. It's a part of the bacterial 70S system. It's part of the 30S. And the function is protein synthesis. We know ribosomes are involved in protein synthesis actually. So the thing with this gene is it has got two types of area. One is a highly conserved area and it has also got a variable area. Conserved area tells you that it belongs to bacteria. So most of the bacteria will share that region. So if you have that sequence, the sequence is coming from a bacteria and not other organism. Then you have the variable region which tells you that, okay, this belongs to this particular genus. It's Escherichia, it is Salmonella. So by variable region, you get an idea probably what that organism is. And by conserved region tells you that this is from bacteria, not from fungi or a human cell or whatever actually. So the 16S rRNA has lot of variable regions and in between there are the conserved region. And you don't have to sequence the entire gene. So what we mostly do is the V3 and V4 region is what we sequence, which is about 300 to 400 base pair long. So now you don't have to do the entire 1500 base pair. Just a 400 base pair, 300 base pair will give you the enough information. So you are covering V3, V4 and the conserved area. So you will know it's a bacteria, you will know what organism it is. The limitation is that you can get a very good idea up to the level of genus. But when it comes to species, the accuracy sort of comes down. So for the species, you may need that whole genome or the shotgun sequencing actually. But for genus level, it's more than 90% accurate actually. So therefore, another way to understand is if you think of a jigsaw puzzle, and if you have one piece of a puzzle, but that puzzle has enough information to tell you which object the puzzle, that piece comes from. So if you only look at that piece, you'll say this is from that object. So 16S RNA is like that. It's your one piece of thing and you know what that is. If you sequence the gene, you know this is the organism it has come from. And this is the most popular technology right now for bacterial study in the microbiome. Next one. So obviously we have loads of organisms, the number of organisms much more than people say than the cells in human body, So the amount of data we generate when we do this sort of studies is massive. You can't be handled by the usual softwares and the tools which we have. It requires specialized methods of data handling and analysis. And these are the terms we commonly come across when we are looking at the microbiome related article in a journal, that what is the diversity, the alpha diversity, how do you compare diversity between the two groups, then how do you identify, like you've done a sequencing, what is that, which organism the sequence belongs to. So there are databases available. So you run it in the database and it will say, okay, this matches with this organism, that's how your bacteria comes. So you identify the taxonomy. There are different databases. You have the green gene and the Silva, which is open source. That's what we use for our work as well. Next slide. And then also for predictors like we do normally, one logistic and multivariate, that sort of a thing is not exactly the same way you do for the usual thing. It is a multivariate but in a different way actually. So here they use more the AI tools like random forest and machine learning things for the analysis actually. And we have specialized pipelines, of which the most popular is the QIIME2, which is the open source. That's what we use for most of our work actually. Because you need these pipelines, the sequencing, which I will come across later. When you get the sequences, you have to run through some pipelines to get an idea what this sort of, the diversity, the organisms, etc. Two things we use is, one we use QIIME2, other is R. R is something, it's an open again software, and you can load a lot of packages in the R software. People have developed this, all free to use actually. So you put the package in this R, and then you can do different types of analysis. You can make plots, you can make graphs actually. So our workhorse right now for data analysis is actually the QIIME2 and the R with all the packages for the data analysis. Next one. That was just an overview of the tools available. So obviously we have the sequencing is the one which is very popular, and a marker gene sequencing is what is more cost effective right now. But I guess with time probably whole genome should be the way to go forward, because it will tell you about the entire, not only the organism but the function as well actually. Okay. You want me to pause or you will continue and then let this thing? Okay, next one. So now we come to the disease part. So that was about the structure, the function, how do we study the microbiome. Now in terms of disease, as I was telling in the beginning of my presentation, a lot of human disease have been now implicated, have been sort of linked to the microbial population in our body, in the GI tract actually. And this just gives you a list, and this again is not a complete list. So for GI tract, obviously Crohn's disease, there is now a lot of evidence to say that microbiome has a very important role in Crohn's disease, ulcerative colitis, colorectal cancer. In liver, it's your fatty liver, alcoholic hepatitis. In fact, already there are trials in our country to show that fecal microbiota transplantation helps with severe alcoholic hepatitis. From Delhi, the ILBS group has done some studies on that. CNS, you have Parkinson's, multiple sclerosis, Alzheimer's, quite a few, bit of papers on this. Obesity, diabetes mellitus, the rheumatological disorder, C. diff infection, etc. So again, if you see, that's a reason, since so many disease processes have been implicated to microbiome, it cut across discipline. It's not only gastroenterology, it's of interest to sort of say medical community in general, and that's why there is so much of interest and research and so many papers being published on the microbiome actually, because almost every disease you think of will be, some microbial link will actually come. And I have not mentioned about pancreatic and also. Sudipto in my department now, he wants to look at the microbiome in pancreatic as well, so it's everything, there is a link to that actually. Next one. So obviously because of time constraint we can't go through all disease, but I thought I'll just cover Crohn's disease because that's what something I've been working on, and I'll take you through the example of how we studied the microbiome from the stool collection till we got the data in the end actually. Next slide. So this project which we had, this was funded by the Department of Science and Technology, Government of India. This was a 4-year project. We looked at 3 things. We looked at the bacterial community which we have in Crohn's patients. Are they stable over a period of time? So one is so we did serial sampling and see if this is a stable community of bacteria. Second is, how are they correlating with biomarkers like calprotectin, CRP, all other markers as well. And also looked at the comparison with controls. Now the control comparison is available from other groups as well, but the first 2 objectives are not really been looked at well. That was the 2 objective. But for the sake of this discussion, I'll just tell you how we compared the microbiome of Crohn's patients with control for the ease of understanding. Now what do we already know about the microbiome in Crohn's? We know that there is a reduced bacterial diversity. This is one finding which has been consistent across all studies, that in Crohn's disease, the microbial diversity is reduced. The actual organisms will keep varying from paper to paper. This again tells you about the diversity of organism and different factors affecting the organism. But among them, the few things which have been observed frequently is reduction of F. plasmodigiae, Clostridia, Bifidobacterium, and things like bacteroids and Rhinococcus are actually increased. So what is the functional implication? So as a result of these changes, what happens is that the short chain fatty acid producers, it becomes lesser, and there is more of a degradation pathway, there is more of abnormal permeability. So overall, this leads to sort of imbalance between there's more pro-inflammatory and less anti-inflammatory microbes in our GI tract. Next one. So essentially the current understanding of IBD or Crohn's is that your dietary factor, environmental factors, the other luminal contents, genetics, they have an effect on the microbial population you have. And the microbial population, environment and your genetic pool together leads to the disease process in Crohn's. But this microbe has now emerged as a very important, you can say a unifying theme in terms of environment and the genetic pool. Genetic pool obviously have a direct effect on your disease as well, but it also has an effect on your bacterial composition in the gut actually. Next one. So for this study, let's say how we went about comparing, say, cases of Crohn's disease with controls. How did we compare the microbial population? So begin with obviously stool collection. Then from the stool sample we extract the DNA. From the DNA we then amplify the 16S RNA, only the area of interest. And then that RNA is read by the machine, the sequence by the NGS machine. And then you use in the database to see what those sequence, which organism that belongs to. And then you do all the comparisons like diversity, etc. and predicted function. Next one. So you take a stool sample. So first step is what we call is a library preparation. Library means you want to have a collection of 16S rRNA which is going to be read by the sequencing machine. So you extract the DNA, you take off the cell wall, other cytoplasmic contents, then the DNA sample, the 16S rRNA is amplified by the PCR. So finally you have a collection of 16S rRNA genes. Now these genes are going to be read by the machine, and that's why this is called a library preparation actually. Okay. Next step. So the sequencing platform, there are a few platforms. What we have in our welcome lab is the Illumina platform. Actually, this is the picture of that. And this, at the same time, we can process up to 300 samples in one go. So what happens is each sample we put some barcoding which is identified for the sample, so that we know that this sequence comes from patient A, B or C. And that way we are able to pool all the data in one. Otherwise you have to run separately. So by labeling the samples, we are able to pool everything in one sort of a thing, and then we can run that in the Illumina platform actually. And in about 48 to 72 hours, you can get the information. Relatively expensive, but cost is coming down. Next one. This is from our lab. So Ira was the person initially started doing this work with me. Now she's an ICMR. Now Dilip is one who is looking after the microbiome work, and we have this lab assistants as well. Next one. So when you do a sequencing run, this is what you get as an output actually. So imagine making head and tails of what is written on this actually, and you get millions like this when you do the run. And that's why you need dedicated softwares and pipelines to say what these belong to. But they have very important information. The first line tells you which patient that sample has come from. Then you have the actual sequence. And then you have the quality. So it's again very important to have quality checks. See, when you do so much of sequencing, there will be some poor quality reads as well. And those poor quality reads have to be filtered out. So while this looks, for a human thing, it looks difficult to read, but machine is able to do it quickly for you. And these are very important. So, and like this, you get millions of reads like this in your output actually. Next one. So that's why this chime pipeline comes handy. So whatever the output we get from our Illumina, we run through this pipeline and it does a lot of things for you without you have to manually do a lot of things. So demultiplexing means it tells you now, it separates out each patient into different categories. Otherwise you have everything pooled together. You want to now separate out the patient's sort of microbiome into separately. Each patient has his own set of microbiome. Quality control, where all the poor quality reads are discarded. And then you make them into operational taxonomy. OTU again is a term very commonly used. OTU means that you put them into one taxonomy, say at the level of phylum in actinobacteria, firmicutes or bacteroids, or you at the level of genus into different groups. So basically operational taxonomic unit is what you put these organisms into after you do the initial analysis. And that OTU table which you get is actually your workhorse for all the downstream analysis. Okay? So you assign taxonomy and you also make a phylogenetic tree based on the relation to each other in terms of evolutionary sort of a pattern. Next one. So as I said, that the operational taxonomic unit table is what is the key from where we can do all the downstream analysis. We can look at the diversity, we can sort of compare the diversity, we can make plots, we can make heat maps from that data which we get actually. Next one. So this diversity is something which is very, when it's like what is the mean age of the population and the gender, this is like that level data. The baseline description of your microbiome is the diversity. And what does diversity mean? Diversity means the different types of organisms, not the number, the different types of organism you have. So if you look at sample 2, there are 2 different colors. There are 2 different types only. But sample 3, there are some 4 different types. So the number of organisms looks similar or less in sample 3, but sample 3 has got a higher diversity. It has more different types. So diversity means the different types of organisms you have actually. And obviously the different types, you can even add the abundance to it, but the more important thing is what are the different types of organisms. So a gut which is more diverse has more different types of species or types of genus or types of organism actually. Next one. So this is our sort of output we get after the initial, the processing in chime. So this tells you that from our sample we got about 13 million reads of those sequences, RNA, and per patient average we got about 50,000 reads per patient actually. Okay, next one. So once we get that, then we want to know what these reads, which taxonomy they belong to. And taxonomy is from phylum, you have class, order, family, genus. Up to genus it is good, species is not very accurate. This is showing you plot at the level of phylum. And each bar represents one individual patient. In the beginning I told that the most dominant phyla in our body are firmicutes and bacteroids and proteobacteria. And those 3 are the most dominant ones in all our patients as expected actually. So you can see the color is basically firmicutes, then bacteroids and proteobacteria. And each thing represents a particular patient actually. So if you want to get a plot at the level of genus, you have to again modify the output and you will get that. But at the level of genus, you will have many more subgroups compared to at the level of phylum, because there will be 50 hundreds of genus actually. Next one. This is another way of looking at the data, but in this, so that one for each individual patient you get 1 bar. If you want to look at the pooled data of the entire set and you want to get an idea about what is happening at the level of phylum, at the level of class, at the level of order, you can get this. This is called a heat tree. So if you see this, At the level of phylum, you can see Firmicutes is the thickest sort of node. So, the Firmicute is the most abundant in your group of Crohn's patient. This is from the Crohn's patient. So, among the Crohn's patient, Firmicutes is most abundant. Then you have the bacteroids and proteobacteria. If you come at the level of class, you see Clostridia is the most abundant actually. So that way you can get an idea what is abundant, because again, since there are so many organisms, you need some simplified way of looking at the whole picture. That's why these plots are sort of made to give you an idea what organisms you have in your sample. Next one. So this is the comparison of diversity of our active Crohn's with the healthy control. So diversity, I told you, looking at what are the different types of species and abundance as well. So as you can see clearly, there are different indices for diversity. This is an example of Shannon diversity. In active Crohn's disease, on the left hand side, you can see diversity is lower compared to healthy control. And we also compare to the stable Crohn's. And in stable Crohn's also you can see the diversity is quite good. So this again definitely proves what other people have found, that in the Crohn's patient, the diversity is significantly reduced compared to controls. And when the Crohn's patients get into remission, when they are stable, the diversity improves actually. And that's what we get information from this analysis. Next one. Then these are the PCOA plots, again very commonly used in any microbiome study. Again, I won't go into details, but with this you can compare between two groups. And there are again complex statistical methods like Permanova, etc., which you can use to see the p-value between the two groups. But PCOA plots are again good, again because of so much of information. These try to simplify your data into manageable, understandable groups actually. Next one. And this tells you the actual organisms seen in terms of the median abundance actually. So the bottom one talks about the median abundance of different genus in active Crohn's and in healthy controls. So if you look at the active Crohn's, the most common is bacteroids, then you have Streptococcus, Prevotella, Faecalibacterium. In healthy control, Prevotella is on top, Faecalibacterium, Streptococcus, bacteroids. And what I said in the beginning about Crohn's is that in Crohn's, Faecalibacterium is reduced, bacteroids is increased. That's exactly what we observed here. You see the bacteroids are on top, Faecalibacterium is low in the active group actually. But again, a word of caution, we cannot just go by this median abundance and say this is more, this is less. So in the microbial sort of a study, a general comparison of median abundance is not very accurate. You have to again use something like a machine learning, like a random forest analysis to say which organism predicts disease, which organism predicts a healthy state. Next one. So this is the example of the random forest analysis we did to finally look at what bacteria are predicting active Crohn's disease, what bacteria are predicting a healthy state. And the dots with different colors tells you that this organism is more common in active, more common or more a predictor of a healthy control. The same information, next slide, can be seen much better on a heat map. So again, heat maps are something very commonly used in your publication of microbiome papers. So heat map, because of the color coding, the darker color is sort of a more relative abundance, lighter color is less abundance actually. So if you look at the bottom one, the Bacteroideaceae more in active and stable Crohn's, less in healthy control, but Bifidobacteria much more in healthy control, lesser in those organs. So like this you can get a visual idea about the different groups, different organisms in different group of patients. Next one. So one limitation of this 16S rRNA is that you don't know about the function. Only a small amount of gene you are sequencing. So you don't know what that bacterial functional capacity is. But now there are softwares which can tell you, which can predict the function based on the 16S RNA, but that an accuracy is about 70%. So not bad, but at the same time not as good. See, finally, our aim is to look at what is the functional impact of the change in the organism. So this is something called the Picrus analysis. Again, it's a little bit technically difficult to do. But again, what we found is that some synthetic functions are better in controls, degradation functions are more in bacteria. But this result, we cannot sort of say it's not very accurate. At the same time, interesting to have an idea about what's going on in among the bacterial population. Next one. Okay. So that was about the, just to give you a flavor of how we study. So we have a stool sample, extract DNA, you amplify 6 RNA. That is read by the machine like the way library books are read. That sequence gives you information to make a table, do the diversity, do the comparisons, do all the analysis actually, okay. Now last few slides on just the clinical applications of microbiome. Next one. So therefore there is a lot of interest in manipulating the microbiota, and these can be done by giving antibiotics. You have the fecal microbiota transplantation, the probiotics. There are dietary therapies which can provide, dietary therapies can provide sort of a substrate for bacteria, which can lead to selective growth of some organism, suppression of some organism. And the precision medicine in cancer I was talking about, that the individuals depending on microbiome, they have different responses to the chemotherapeutic drugs. So for antibiotic, we have example of SBP, where somebody gets SBP or spontaneous bacterial peritonitis, they are on prophylaxis with antibiotic long term after that. For Clostridium difficile, it's a well-established therapy. Salvage therapy is fecal matter transplantation. Probiotics have some role in pouchitis and traveler's diarrhea. The diet therapy for IBS we frequently use is low FODMAP diet in people who have not responded, and this is supposed to work at the level of microbiome. Next one. So just briefly about the IBD. So among IBD, there is more information on ulcerative colitis. Next one. Compared to Crohn's. So Crohn's, the data is still emerging, but for ulcerative colitis, this is a well-established therapy based on RCTs. But the problem with FMT is that it's not very acceptable to the patient. I mean, somebody says that I'll give you somebody else's tool, they will probably be put off with that idea actually. And also we have a lot of therapies available. It's not that we are really struggling with therapy. So there are a lot of other therapeutic choices in UC. This is not very acceptable. That's why it's not taken off. And also the logistics of storing the stool, screening the donor actually, doing the whole procedure. It's not a one-time procedure. You have to keep doing repeatedly. Otherwise, after you do the transplantation, soon again the earlier bacteria comes back after some time. So it's not a one-time procedure. So the logistics of doing it, that's why it's not as popular a technique as of now. In India, too few centers are doing it. I think in Ludhiana and AIIMS New Delhi they're doing it, but we are not doing it. And that's because in colitis, we do have therapy, Misacol or Azoran or something which is quite okay. But this is something, if it becomes simpler, if you have a capsule or something which you can take, it will become more this thing. And there is now efforts to develop capsules rather than doing all this cumbersome colonoscopy and putting stool sample inside the colon actually. Next one. Just to give you an idea about the amount of interest there is in terms of bacteria-based therapy, more than 25 things are right now in development, which is all targeting your microbiome population or using microbiome for therapy. Next one. So for the oncology people, again I said now the human microbiome and the personalized cancer treatment, especially for the checkpoint inhibitors, what I said, some people respond, some do not respond based on the type of microbial they have. And for them, this is again a big area of research and see how they can pre-identify which patient will respond to this drug, which patient will not respond to drug. Next one. Okay, so this is the last one again. So just to show this picture, you're seeing somebody, so his hair has not grown because of hair transplantation, but this is because of fecal microbiota transplantation. So this is a patient who had alopecia areata, and he had C. diff infection, and then he got FMT, and then as a collateral benefit, he also got his hair back as well, So obviously this is because of the alopecia areata had this immune-mediated effect actually. So this probably had some impact on the immune system in the GI tract, and now there are at least 3-4 case reports of this happening. So it's not the only case report we have actually, okay. Next one. Okay, so my last 2 slides now. So far I've been talking all good about microbiome and what you can get and all that, the disease, everything. But it's very important to put things into a perspective. One is, in most situations, we don't know about the cause and effect, Like whether the organism changes happened because of disease or the organism is causing disease is not very clear. So until you have, then that's where the long-term study or the longitudinal study is important. So you study longitudinally, you look at the microbiome, and then you can priori have an idea what the microbiota was. Second thing is that organisms live in a community, so we can talk about individual bacteria and talk about their functions. But when they are together with so many organisms, we don't know how their function is changing and what they're doing at that time. Also, as I told you, that there are so many factors, diet, antibiotics, your geographical location, to find out what is causing the changes also. I mean, what sort of, to filter out the effect of these factors. And then to get only the disease causing pathogens is also not very easy. And despite all the progress we made, still we have about 10 to 15% of the organisms which we don't know what those organisms are actually. And obviously we've been all generally focused on bacteria, but what about the virus and the fungi and everything? Next slide. So in conclusion, again, understanding the structure and function of microbiota is an evolving field. It's an area of significant research interest, as I showed you with the number of publications. It has very important role in development and function, the immune system, the nervous system, etc. It's been implicated in pathogenesis of a lot of disease process, and manipulation of this bacterial community is emerging as a therapeutic modality for several conditions. But at the same time, despite of all the progress we have made, I think our knowledge is still quite limited, especially about the functional and the metabolic potentials of the microbiome. Thank you. Yeah. And it was a long topic, but I know this is like we had to cover this much to get an idea about this whole thing actually. Yeah. No, I think it's good to know this topic because this in future you'll have more and more papers, and as it cuts across discipline. So finally, even when you read a literature on microbiome, at least in your mind, you should know what these terms are meaning, the diversity, the plots they're drawing, how they have looked at the predictors and everything, and what actually goes through the limitations. All that is good to have a thing. And it takes a little bit time to understand, but if you read more and more articles, then it starts getting clearer in your mind about the microbiome actually. Yeah, so any question, anybody, I'm happy to answer. All over the top of head, I think. Yeah.
Thank you very much for your presentation. The microbiome is one of my areas of that I like to explore and learn more about all the time.
I can't hear you.
Question number one.
Yeah.
You've also mentioned, and in the literature we see that there are many, if I can call them, risk factors, which includes antibiotic medication use, geographic location, early infant feeding, mode of delivery, etc., diet, activity, etc., etc., etc. Have we, typically it seems like we mention that and leave it, and then focus on the microbiome. Have there been any efforts to try and do, this is probably the wrong terminology, any risk factor scoring for unhealthy alterations of the microbiome, and combine that together with microbiome analysis and see those that have more risk factors, or certain risk factors are more prone to more severe disease, etc. Is that something that is being looked at all? And number one. Number two, as you mentioned with CUC, those people that are getting fecal transplants, it needs to be repetitive and more than one. We also see it with obesity and other areas that are emerging, as you have suggested. If one were to look at what are some of the quote-unquote risk factors or lifestyle risk factors that the donor didn't have, or the positive lifestyle components, or the positive factors that the donor has, which the recipient doesn't have, if the recipient would then adopt some of those, would that mean that there may have to be less frequent repeat transplants?
That's very, very, very interesting set of couple of questions. The first question was mainly, which is what the important thing is, that when you're studying a disease process in microbiome, how do you filter out or at least know the effects of different things actually? So there are statistical methods actually. One is that, like the way you do a multivariate in a normal statistics, there are analysis possible where you can adjust for factors actually. So there are some pipelines available as well. So that can tell you, and it can filter out the effect of those factors, but you cannot filter out too many factors in that. So what we people tend to do is they may have a subset of patients who are a bit more homogeneous in terms of other factors, so that there is adjustment, like you choose from one location, you don't choose from all over the place. You make sure people have not got any antibiotic in the last 3 months. So you make sure you choose sort of age matched. If you're doing a control, do an age matched control actually. So you do some factors for adjustment priori before you recruit the patient. And second thing is in analysis, you can do some multivariate analysis as well, which is possible. I think there are some pipelines. There's one Maslin pipeline which is available, where it can adjust few of the factors and you can get. And also the PCOA plots which I showed you actually, the principal component analysis, that can actually tell you the contribution of different things to the microbial change. So when it compares the two groups, it will tell you what was the impact of age on that group, what was the impact of disease on that group, what was the impact of this thing. So what we have found in studies in Crohn's is that actually if you look at the two group comparison, the maximum impact is coming from the location and other things, and the disease probably ranks third or fourth in the list actually. So there are ways of finding out the actual impact of, then you can put some number, some percentage to know that okay, the disease itself is accounting for 10% change in microbiome in the two group, but 80% of change is because of the location or the gender or the thing. So that is possible too, but these are again, it's like the data is so vast. So we try to reduce dimensions by this PCO analysis, and then this is what is called as a dimensionality reduction by this principal component analysis. And then we look at the first component and you see, okay, this component which is the most important, this much was contributed by your, say, location or your diet or your age, and this was contributed. So that's where we found that disease process does contribute, but in some disease maybe a lower contribution actually. So there are some analysis methods to look at that, but you also try to adjust priori before the study for the factors so that you have, like for our study, it normally initially focused on people from this part of the country. We have matched the controls with the where they come from, as well the age and the gender we have matched. We have not taken any I think we've hardly had anybody with cesarean section delivery. So those things, whatever, and we have got a very sort of, detailed dietary record, the 3-day recall of the diet, so that we know that for that. The thing is, so you can adjust for some factors in that. I think the second part of your question was, can you just repeat the what was your second question? Second question was, you asked the second question, isn't it?
If one were to look at the positive lifestyle factors, would that reduce the number of...
Yeah, so for the fecal microbiota transplantation, right now if you look at the FMT, there is a stringent criteria for selecting the donor actually. So probably somebody obesity will get excluded in that thing, you know what I would mean. So essentially there is a very stringent criteria for donor selection, which also includes the looking at the stool for pathogens by a PCR panel and other methods actually. And at the same time, all the comorbid like diabetes, hypertension, any other chronic illnesses, inflammatory disorders, all those we have to exclude before we put them as a donor. So donors are generally healthy young adults in that group actually. And then their stool is actually screened. And most places it's gone to the frozen sample, because real time you can't get a donor immediately when you need to give it to somebody. So you take the stool sample, you bank it. In fact, I think in Hong Kong or in some country, you get paid if you give your stool sample, So it's like, you give your stool sample, so healthy donors who give sample get paid to contribute to the stool bank in that place. So I think in Hong Kong or somewhere it is happening like that actually. So that's one thing. Second thing is, despite of all these things you do, like this ulcerative colitis studies, what they have found is there are some donors whose stool is very good and doesn't depend on their obesity or what are other things actually. So these are called super donors actually. So what they found is there are some donors, when samples goes from them, those patients do very well. So this phenomenon of super donor is also being identified. So you'll find out that some patients actually are very good in terms of so when their thing is transplanted, the effects are good actually. But the reason for that phenomenon is not clear, because these are generally all similar in terms of their health and comorbidity, hardly any comorbidity actually. That's what I think the question was actually, yeah.
And thirdly, and I don't know if you're already doing this or not, but do we have any RCTs looking at, for example, Crohn's disease? Here's our standard of care. This is what their microbiome looks like. And then another group, or the intervention group, the standard of care would be the control group in this case, and the intervention group then would be, if we give intensive lifestyle, and maybe we have weighted some of the factors that are most relevant and most important, how do these people compare with, versus standard of care? And then how do these people compare with standard of care versus intensive lifestyle, however that is measured?
Yeah, definitely. So I think I'm not aware of any RCT so far, but these are things now people are different. So now if you look at a lot of studies on IBD globally, microbiome is becoming one component of it actually. So you also collect some stool sample for microbiota to look at how that has changed. But as of now, there's no RCT from what I can recall in terms of the lifestyle interventions and the other group to see how it changes. But there are, in fact, for example, now we are looking in our gastro, like one of the theses is we're looking at anemia is very common in Crohn's disease. Some guys respond to oral, some guys don't respond, some guys need a parenteral injection actually. So we are looking at the response to oral iron therapy, and we're also looking at how the microbiome is sort of difference between the two patients actually. But see, these again, the question sounds interesting, but there won't be as much information as we think it will come because of the limitations which I have mentioned. But I think you have to start from somewhere, You cannot also expect to get everything in one go. So you have to start from somewhere actually. So it's becoming, so microbiome thing is now becoming a part of a lot of these research where they're using Crohn's and everything actually.
So in Crohn's disease, CUC probably is a better example. If we have an idea as to what proportion of patients respond to our standard of care,
yes.
And then if we take another group and say what proportion respond to intensive lifestyle or moderate lifestyle intervention, is there overlap? Is this a small subset? Is this fairly general that we do see significant improvements simply with lifestyle, without the pharmacologics that we add on? This is an area that would also be interesting, because there's a lot in the literature individually, but I'm not sure there's much comparing the two.
Not much. This is an excellent suggestion. In fact, so this I told you, now the focus is more on a serial study. Looking at one point may not be very, informative. So like even for the project which we did, we looked at 2-3 time points. Obviously, because of the COVID coming right in the middle, some follow-ups were difficult, but still we sort of had research calling these guys, and we had some follow-up actually. Even yesterday, I did one IRB presentation, or day before yesterday, about we are looking at these patients who have active Crohn's disease. Now they have gone into remission. We want to see how the microbiome has actually changed in them, So now we have that for the project, we had 60 patients who had active disease. And now we want to take this tool and see how things have changed after they have gone into remission. So these serial studies are probably going to be more informative. After change in the disease behavior, as you said, how things change also will be more informative. But then that will give you some information. You have to build up on that actually. Okay.
Thank you very much. We look forward to collaboration more in the future.
No, thank you. So any questions from the residents? Anybody wants to ask any question?
Thanks, Amit, for this beautiful. I think it's opening our eyes. One thing that comes to my mind is, as you said, whether it's a cause or an effect, transplanting feces is one thing. If we can build up that spectrum in our own body through other alternative way would be something, looking at from a lifestyle perspective, is if we can get that healthy microbiome in our body through a healthy lifestyle. Because we are also seeing about mental issues affecting the microbiome. So almost virtually our microbiome could be a reflection of what's happening in our life in different domains. And if we have that, so microbiome is helping us understand many things. So if we understand that and understand that factors that gives us a healthy microbiome, so instead of going for the feces and putting it, which is the easiest thing to do, maybe it may make more prudence that we change those things so that our microbiome changes, and thereby we achieve health on different aspects of disease. So I think it's very exciting, and thanks for clarifying all those terminologies and the perspectives and the complexity of the whole thing, so that people in future would be working more and more in that area.
In fact, one more point on FMT I want to add. So look at the both aspects. Now there have been reports of MDR bacteria being transmitted through this FMT as well. So so you have drug resistance as a. So in fact now there is a call for screening of some of the bacteria in the gut before you actually So everybody with MDR may not be having the symptom. So there have been, I think there's a report of death as well, if somebody got that. We look at the whole perspective, not just a one-sided view actually. So FMT, that's the thing we have to look at. So as you said, all the things have to,
One more thing is coming is the microbiome, or rather microbiome basically means the spectrum of different organisms that you have, would also be a very individualized thing.
Yes.
What you develop a tolerance to and what you get rid of is very individualized, and depends on what you were earlier exposed to and what happens through different events of life. So simply taking up a microbiome of one person and putting in itself may be, we have to be very cautious about, because it has to be some kind of a gradual process that may have, and our body may be used to a particular thing, and so suddenly giving them something new, we don't know what is the wisdom in that, but something that we'll know more and more over a period of time, but something that we need to also be cognizant about.
That's why we are not jumping on the bandwagon of FMT so quickly. We are sort of taking the time. There's no dire need except for C. diff, which we are not seeing much, but now 1 or 2 cases we have seen. But earlier, CMC, luckily in India, we don't have as much of C. diff as they have in the West, but maybe things will change with time. So, that's one area where we may have to. But for IBD, as of now, we are okay, but we'll develop slowly, I think.
Lastly, there are some areas like obesity, which studies have shown that if I happen to be in a family with an obese person, I have an increased likelihood of also becoming obese. If I have friends who are obese, I have an increasing likelihood. I don't know how well that some of those associations have been made with other diseases, diabetes, hypertension, and so forth. But the question comes in, is that association more from other additional lifestyle factors, or is there some type of microbiome transference that happens with that association, which helps with the increased incidence?
That's again a very important, very interesting, and I think there is data on that actually. So they have done studies on germ-free mice, and they have taken microbiome from obese mice and put it in the thing, and so they have, and then they have taken microbiome from lean mice. Amount of food they got is same. The one from obese mice became obese, one from this thing was lean. So essentially what they say is that your calorie extraction from the diet, Bacteria also extracts a bit of your calorie, and that is sometimes the thing. So basically, the people who are having obese may also have a bacteria which is very efficient in extracting the energy, and then which the human body uses, and the bacteria also uses. So that's why I think the joke goes that if your friends are obese, you also will be obese. Probably I think there is some transfer of microbiome. What people feel is they're happening actually. So that's why obesity again, it's not only that people say, I'm not eating much, but I am gaining a lot of weight. Often you'll hear that in your OPD, I'm eating very little, but I know how I'm putting so much of weight. So there is definitely microbiome in obesity. There's implication, and the studies in mice have shown it actually. Yeah. Tambu?
I'm very happy to hear that. So, guys, this is not what I'm eating. It's all the bugs in my stomach. So,
so this may be in future. There may not be any more non-communicable disease. You know what I mean? The classical NCDs of hypertension, diabetes, obesity may soon become your communicable disease. Microbiome coming into picture actually.
fecal microbiota for everybody. Let's give Amit one more hand. Thank you, Amit.